Hey everyone. Yeah, as said before, I'm Aidan and this is Volkan. And today we're going to be talking about our research on using synthetic data for training deep learning models to process time-lapse microscopy of mitochondria, and also about some of the insights we gained from analyzing this data. This research has come from a long-term collaboration we at the CLSMB lab have at Melbourne have with Dr. Senthil Ramagam's lab at Monash Biomedical Discovery Institute. They're responsible for the amazing data that this project uses. One of CLSMB's long-term focuses has been on creating multi-scale biophysical models and digital twins of cells. Mitochondria are a particularly interesting component in this modeling as they're one of the centerpieces of cellular life. Beyond making ATP, mitochondria help control calcium, lipid metabolisms, and signaling through reactive oxygen species. They also regulate programmed cell death, which matters in cancer, neurodegeneration, and development. More recently, they're being recognized as signaling hubs that sense metabolic stress and help trigger immune responses. A lot of existing literature in this area misses a critical point. None of this happens in a fixed static structure. It is not localized to a single mitochondria, and they change and move and interact over time. The role this project plays in this bigger picture is using image processing to extract both static and dynamic insights from mitochondrial microscopy data, and in our case, help create better models with the behaviors and properties observed. The first challenge in processing mitochondria data is that they're quite diverse in shape and appearance. They range from tiny puncta and tubules all the way to big branching structures and densely connected networks. Importantly, this range can occur within a single cell, either spatially across disparate mitochondrial regions or temporally as structures change in response to internal or external stimuli. Sec. The second challenge is in quantifying the dynamics to give us better insights on how they change both in morphology and cell-wide arrangement. These take the form of both long-term migration events that happen over minutes and hours, and local dynamic events like fissions or fusions, which happen at the scale of seconds. This is all to say that dynamics and morphology can't be considered separately, which is why there is a real need for simultaneous analysis. For example, the impact of modulation of fusion and fission events on longer-term emergent network structures and properties remains unclear to this day. This brings us to the data created by Senthil's lab that is going to make this desired analysis at all possible. Their setup captures images for 3+ hours at 2 captures a second, with a field of view wide enough to capture a whole cell, even through cellular migration, and with spatial and temporal resolutions high enough to resolve individual mitochondria during fission and fusion. This is possible due to their protocol that keeps laser power low to avoid photobleaching from the repeated exposures over such a long duration. Just hit play on these. And here is a non-photogentle sample from that same setup to get a clear picture of the size and scale that we're dealing with. But I don't think it played. For those of you more technically inclined on the microscopy front, which isn't us, unfortunately, the microscope was a Nikon TA2E with a 60x objective. If you have any extra questions on this, it's probably best to send Senthil an email and he can probably answer. So, to learn everything we want to, we need to do one of the harder kinds of image processing, which is the combination of instance segmentation and object tracking. This is also called video instance segmentation or VIS or VIS in other fields. This combines the per frame separation of individual entities in instance segmentation, which is in our case mitochondria, with the intra-frame matching to know what entities become what in the sequential frames. The aforementioned fission and fusion does make this slightly harder than a standard framing in other fields. For example, in self-driving cars, you don't have one person turning into two in the next frame. while you have situations like the one in the bottom right of this figure, where the mitochondria in yellow turns into both the ones in pink and green in the next frame. You can see this. I don't know if you can see my mouse, but through here. Two papers that explicitly tackle this challenge are Mitometer and Nelly, both by Lefebvre et al. Both works use a two-step segment and then track approach. So this means that first they use algorithmic segmentation to create candidate instant masks from which they can extract visual and dynamic features. Then they use a matching scheme to assign end-to-end instance continuity, detect fissions and fusions, and clean up erroneous masks. Unfortunately, for our photogentle data, their approach doesn't work. The low wattage illumination required to stop photo bleaching causes a very low signal to noise ratio. And their first step, algorithmic segmentation, produces low quality masks. With both feature extraction and tracking downstream of these masks, errors quickly proliferate and it creates an unusable output. Deep learning models seem like the obvious choice here, as they're state of the art in most other computer vision fields, and as they are more robust when noisy data is a problem. Deep learning model also has some added benefits. It could see multiple frames at once, therefore not relying just on the data from the current frame being segmented, and instead use temporal cues. They also have potential to explicitly learn dynamic interactions, like if there is a difference in motion between two mitochondria just touching and those undergoing a fusion event. There has been extensive research into video instance segmentation deep learning models for tasks like self-driving cars and automation of video surveillance tools. There is also some more relevant literature showing these models excel at whole cell tracking, so with papers like Cell Tractor, but none of these explicitly work for the end-to-end interframe matchings we have for fission and fusions. And therefore, and furthermore, the bounding box methods that they most commonly rely on are not suitable for long intertwined objects or network-like structures that we have. So we needed to adapt or create our own model. The first roadblock we almost ran into immediately was creating a training data set. It has a very high upfront cost in all problems, and we had no guarantee any model we would create would work. It also is not an easy annotation task in the first place. Some domain expertise was going to be needed to properly separate out individual instances, and creating consistent masks across multiple frames is extremely time consuming. Our solution to this was to use synthetic data as a placeholder. It meant we could quickly work out if any approach we tried was just bad or it just didn't have enough training data and still could potentially work. It also generates perfect annotations every time, and annotations can be adjusted programmatically for different training targets. For example, we could change the labels if we want to train a model just on semantic segmentation, or we could swap it all to classify punctate versus tubular mitochondria. Other fields have shown this as an effective method, such as studies pre-training self-driving cars in video games and the NVIDIA Omniverse platform that was created explicitly for training robotic automation in simulated environments. There is also some more domain-relevant literature, like a paper called SynSeg, which renders synthetic microscopy stills of lysosomes, and a work by Prasad et al. That does the same for tubular mitochondria and vesicles. This brings us to MitoMimics, which is the tool we created for this purpose. It makes realistic enough time-lapse microscopy data that a model trained only on this data achieves state-of-the-art segmentation and tracking performance. We create these time lapses in a two-step process. First, the morphology and dynamics are created with a stick and ball model, a technique used in mitochondria modeling literature to represent their arbitrary network structures. This gives us a very simple way to achieve dynamics through just rigid bodies and simple forces, while allowing for relatively complex behaviors like fission, fusion, and which are just the addition or removal of the stick link constraints. To make data match as close as possible at the whole cell level, we also implemented per instance behavior states, migration, clustering of networks around the nucleus region, and cell-wide event regulation. We manually adjusted the knobs that change these features to roughly match those observed in the real samples. The second step is rendering these into images that look like they actually came out of a microscope. For this, we built a multi-stage Python pipeline that stacks noise in a semi-realistic way. It uses approximations of shot, dark, and quantization noise that make up the signal portion of the image, with a background of Gaussian noise and random blobs to represent arbitrary structures and debris. The values of the parameters were tuned by starting from the real-life baselines when possible and adjusting manually to match noise curves and visual appearance. One side benefit of this approach is we get a free additional form of data augmentation to increase the robustness of our model. Data augmentation is a technique used when training deep learning models where images are algorithmically altered with things like brightness, contrast, added noise, etc., to increase robustness and generalization. In our case, by randomly changing the rendering parameters around their tuned values, our synthetic dataset can have a high level of diversity, but still more realistic than just standard data augmentation methods.
So just a
Oh,
question.
yes.
So the slide where you talk about using Pygame, right, for simulating the mitochondria, you said it's similar to the real data. Like, how are you going to talk about how physics constrained or how similar this is to that, or are you going to talk about it, or maybe I'm just asking earlier?
it, it's not in the scope of this presentation. It is a very much a mock simulation. Like it's not trying to be a true simulation. It is trying to match what we see visually, and also to match the overall rates we see, like observed for fissions and fusions, and how quickly they translate across the field of view, stuff like that. But their actual forces used aren't realistic.
Oh yeah, maybe not that. Maybe I use physics constraint quite loosely there.
Yeah.
So I guess what I'm trying to get at is maybe more biologically relevant is the right term. So what I mean by is, I think you answered the question. It's like the fission fusion rates, the all that. So you have the information for that and the spread of
Mm.
it. And is that what you use in the model? Is that uh,
Yeah,
con-
the values are in the model because it's sort of an agent-based. The mitochondria are given individual states, and those states were balanced so that it fell into the rough range that we would expect in real images.
okay? All right. So yeah, okay. So each of them are individual entities or so.
Yes.
Yeah.
Yeah.
Okay. All right. Yeah. Thanks for answering.
And here's some comparison stills with different noise profiles, with MitoMimics on the bottom and real images on the top. And here is a side by side at real-time playback speed. The actual model we ended up creating was a hybrid of the segment and then track approach. Semantic mask was still created first, but unlike Nelly and Mitometer, they segmented multiple time steps in a single pass, with the 3D model we used treating time as the third spatial dimension. We did this by training on 64 frame patches that were randomly sampled each epoch from the full 2000 pixel square volumes that MitoMimics creates. The total dataset ended up with 256 volumes and took around 36 hours to train on Spartan, which is Uni Melbourne's compute cluster. To separate instances from the semantic masks, we trained a second centerline only model on the same 3D temporal stacks. Unlike with the first model, which segments the whole morphology, in this model, two centerlines from separate instances would never touch unless a fusion event occurs, and they would never split unless a fission event occurs. This allowed us to build a post-processing algorithm that determines separation from the centerlines, instant separation from the centerlines, and produces an end-to-end interframe matching from their 3D continuity. We then use those centerlines to watershed the full masks with instance identities and get the final tracked output. We can see down the bottom of this figure that the yellow mitochondria is detected as having a fission into the pink and green mitochondria in the next frame, but before its full morphological masks have separated.
Right, just with the previous one. Uh, so the previous slide. So essentially, it's like you have the skeleton, and then you
Mhm.
have the entire mask. Those are the two
Yep.
things. Okay, all right.
Yep.
Just make sure I understand correctly. Because when I say center line, yeah, I just assumed it was
Yeah.
a centroid, but you had center line, so that makes sense. Yeah,
Yes.
cool.
Yeah. We changed name midway through the project, calling them skeletons to centerline, so we would get less confused.
Okay. All right, cool.
Um, as the model is just a 3D model at its core, inference for longer durations or bigger samples is as simple as just turning real time lapses into big volumes and using the NNUNet framework UMamba is built onto to do the rest. This works by doing inference on small patches, the size the model is trained on, and sliding them to span all of X, Y, and T. Then stitching the predictions into big stacks of masks, which are then post-processed into the final outputs. In this video, we've overlaid track segmentation on the real data. The color changes you're seeing when fission or fusion occurs, or when a new mitochondria appears either from being occluded or coming into focus. To validate this approach, we actually do the thing we were avoiding in the first place, which is annotate some real data. We annotated 48 small regions from still frames where every mitochondria is given its own ID, and adjacent frames we use to better separate instances in particular noisy areas. And for tracking evaluation, we labeled 60 track sequences of specific mitochondria or plural across 48 frames. This was done in Apari with some added hotkeys to quickly swap between frames to better visualize changes between frames. As far as performance goes, the improvement over the baseline methods is massive. MitoMimics isn't prone to the same oversplitting of single instances like Nelly is, or the inability to create accurate morphological shapes like Automata. For numerical evaluation, we use the go-to instance segmentation metric, which is mean average precision. Simply put, for each prediction, check if it crosses an intersection over union threshold with the ground truth instance. If it does, mark it as a true prediction, and repeat this for all mitochondria predictions at 10 different IOU thresholds, with the final point metric being the average of the precision of these predictions at each threshold. This gives more of a holistic view than a single threshold or point metric and can be useful if you're willing to trade off mask accuracy for object prediction accuracy. Um, for analyzing tracking performance, we categorize dynamic events into 5 categories. We had control events where mitochondria did nothing of interest. We had true fusion events, and they're commonly confused near-miss events, where two mitochondria would be close for an extended period of time, but without actually fusing. We also had fission events and what we termed low signal events, where a portion of a mitochondria dims or is noisy enough that when labeling, they could be confused with real fission events. To count as getting an event correct, the model had to have sufficiently high overlap with all the mitochondria in the event across all time lapses, or across all the time lapse, sorry. And then it had to report the same event within 2 seconds of what it is labeled as. And this is a big stitching of a 3 hour movie at 200x speed we created with it. And to talk about what we actually found out about from this data, I'm going to hand you off to Volkan for the rest of the talk.
All right, amazing. Thanks, Aidan. So to quantify these emergent properties, we measured how dynamics really remodels mitochondrial network topology, connectivity, as well as spatial reach. So how these networks evolve and interact over time. And so using the MitoMimics segmentation and the analysis pipeline, we can extract features from raw image stacks, as shown here. And there's multiple levels of analysis you can conduct. So there's instance level object segmentations, which Aidan showed. There's also branch level subsegmentations we can do, network graph structures, as well as node positions. So here in red are branch nodes, and in green are endpoint nodes, which allow you to show and depict the evolution of networks over time. Here, I'll just focus on instance level temporal features, which reflect the direct consequences of fission fusion activity. And so as MitoMimics has been created to expand the segmentation across varied signal-to-noise ratios, we wanted to continue studying the effects of photo-induced damage on mitochondrial dynamics. And in these images, we've used the heaviest irradiation levels just to depict these. And we see that there's a gradual change in morphology and dynamics in mitochondria. And so they transition from more network-like tubular structures to fragmented puncta and smaller tubules until the cell ceases and undergoes apoptosis and just dies. And this is a video across the different ranges of light dosages that we used in the experiments. And we see that it can be quite difficult to see if subtle changes are occurring in dynamics. You can see from the previous slide that there are changes at the start and the end, but the ongoing changes are also very important. And so by benchmarking these heavily damaged states, we can identify and eliminate any of those artificial stresses that may compound effects of mitochondria, such as those stress-dependent fission and fusion, as well as sustained degradation. And so you can see here in the lower end of light dosages, by the time it reaches the end, their networks are still quite tubular and well-connected, while at those extreme stressed ends, they form more puncta-like structures. And in addition, we can use the segmentations and those temporal tracks of fission and fusion to measure genuine quantitative changes in populations. Additionally, another aspect over just irradiation levels that we wanted to look at was two functionally and morphologically distinct subpopulations of mitochondria. So usually in cells, in certain types of cells especially, you can get two populations known as perinuclear and telenuclear mitochondria. Other cell types can have different subpopulations as well as more fragmented morphologies. And so with perinuclear mitochondria, which is shown in the yellow border, they're clustered tightly around the nuclear envelope and form these highly dense interconnected networks, while telenuclear mitochondria are located towards the outer edges of the cell, the periphery, and generally are more tubular, at least in the cell types we were using, which were retinal pigment epithelial cells. And the cell constantly traffics mitochondria between these two compartments. So with our high accuracy segmentations and those long sub-second resolution imaging, we were interested in seeing whether there were differences in dynamics, particularly fission and fusion, between these subpopulations, as well as comparing that to the global total population. As usually in the literature, we found that only the global metrics are actually reported, and it doesn't get to the why or the how of spatial subpopulations. And so the first bit of analysis that we did with these lovely segmentations was measuring the cumulative event differences over time of fission and fusion events. And so what we're doing is we're taking the counts of fission and fusion events every half second, and then we're cumulatively stacking them and then subtracting fusion events from fission events to see how those subtle changes occur over a 27 or so minute time window. And so in the high intensity conditions, which is in the two darkest purples, we can see sigmoidal accumulation kinetics of fission events after approximately like 5 to 10 minutes of sustained irradiation damage. And this occurs in telenuclear and perinuclear as well as the global. On the other hand, the low intensity conditions in the lightest two pink colors, which are at the bottom here, showed a very near constant, or what we can call like a weakly saturating behavior. We then wanted to fit sigmoids to this, so we used a 4-parameter logistic regression fits and mapped their inflection points to yield what we would call as a global damage index. And this global damage index, which we've created, measures photo-induced damage stress, which reflects the rate of divergence of fission and fusion event counts that we see here. And so a damage index of less than 0, so here, indicates that fusion outweighs fission events. So the system shifts towards elongation and repair. A damage index greater than 0 in these higher irradiation conditions indicates fission events outweigh fusion events, and the system likely shifts towards damage-induced fragmentation and stress-dependent fragmentation. And a damage index of 0 indicates no bias towards fission or fusion. And so if we fit a linear trend line to this, we can identify a transition-dominated fission accumulation, which occurs above approximately 28 milliwatts per centimeter squared. And this indicates to us that the ideal safe zone for imaging is likely around the 10 to 28 milliwatts per centimeter squared. This is also such that we don't have impairment of mitochondrial dynamics over the long imaging periods we want to show. And again, this is seldom reported in the literature, as usually most of the experiments are done over, say, 5 minutes or so. And so on top of this, the next stage was looking at those actual fission and fusion rates. So across each condition, we quantified fission and fusion rates as overall averages, so using the averages across the entire time window, as well as time result profiles down here. So looking at the fission and fusion rates over time. And so the aggregate metrics indicated that fission and fusion rates were comparable and they look very similar under photogentle imaging, while higher illumination conditions drove quite a marked increase in both processes, so in both fission and fusion. However, while these averages are normally reported in the literature, the temporal trends are very rarely ever actually reported. And so looking at these temporal profiles, they reveal a high dose condition rapidly increases the fission and fusion rates. And in different spatial compartments, those rates differ quite dramatically. On the other hand, the photogentle samples show constant or stable trajectories. And this is highlighted to us that the aggregate rates, while useful for overall values, appear to actually mask quite a lot of the temporal dynamics associated with mitochondrial homeostasis and quality control mechanisms. So interestingly, the rates, again, for fission and fusion appear very similar. So we sought to try to disentangle some meaning out of this. And as they appeared similar, we wondered whether there was a correlation between the two. So we use a cross-correlation analysis, which measures the similarity between two different signals as they shift relative to one another. So we use those time-resolved profiles for fission and fusion, and we wanted to see whether this cross-correlation analysis could tell us something about those shifts. And so it tells us how well two patterns match and whether one pattern leads or lags behind the other. So here, does fission or fusion come first? We took the fission and fusion time trends and linearly detrended these to remove any artificial effects. And the signals were actually standardized by z-score normalization for comparison between conditions. And so across each irradiation level and spatial context, cross-correlation analysis revealed synchronous coupling of fission and fusion events as seen by the strong zero lag peaks. There doesn't appear, at least from this data, to be any consistent leader-follower relationship. And what we think is that therefore there's a small imbalances trigger very immediate and synchronous fission-fusion coupling to preserve cellular mitochondrial morphology and function and to relieve the cell of stress. And so just summarizing that, we find that overall the standard rates of fission and fusion in the literature are are likely not sufficient to drive overall mitochondrial fragmentation, and that there might be other mechanisms at play here. And so fission and fusion processes may play a secondary role or an ad hoc role in structural network changes compared to other cellular mechanisms. And these processes may not be the primary regulators of mitochondrial breakdown during stress-related responses. And these really need to be reassessed in the literature. Additionally, we've shown these for phototoxicity examples. However, we've also conducted the rates comparisons and network spreading for protein knockouts, so more interesting biological conditions. But for the scope of today's presentation, I won't talk about those as that's quite a different story. But we do observe similar fragmentation and structural behavior occurring under direct genetic manipulation, suggesting alternative drivers may be at work. And so here we have one of the knockout conditions that we've used, which is a membrane curvature protein that also localizes on mitochondria. And we've compared that to the wild type. And under these steady state conditions of genetic perturbation, they form more fragmented networks, and we continue to study those. And so just coming back full circle, a major takeaway from this work was that we ultimately never really needed to create the large hand-labeled datasets that MitoMimics was originally designed to replace. The generalization from synthetic training data to real microscopy data actually far exceeded our expectations from the start of this project. And the scale of the data used to train the models would have been quite impractical and very, very expensive to annotate manually. I'm sure we'd need many people doing this day and night just to get this level of segmentation. And so we believe that this approach could extend to many other microscopy and biological imaging applications where image characteristics and morphology are sufficiently well understood or can be closely approximated to realistically simulate those. In terms of limitations, overlapping mitochondria in 2D imaging can occasionally be misclassified as fusion and subsequent fission events. However, this is largely an inherent limitation of 2D analysis rather than the pipeline itself, and is actually shared by all the other existing segmentation methods, regardless of what you do. While we could go to 3D time-lapse imaging, and that could address the issue, it introduces additional technical and experimental challenges. And so finally, when we're comparing this to other literature for segmentation, there is a trade-off between performance and usability. So tools like Nelly and Mitometer offer a very accessible one-click workflow, which is preferable if you don't like coding or you don't have a lot of data, stills maybe, or if you just have clean data sets that you want to segment and look at. And so the performance gap might be quite small and you might not get those over or under segmentations. However, the data sets which are not perfectly clean, so like the data sets we showed over long several hours sub-second imaging on a low-cost imaging modality, there is going to be massive accuracy trade-offs in performance in these tools, as these other segmentation workflows are purely traditional image processing algorithmic workflows. And so finally, we believe that one of the most promising future directions for this approach is obviously 3D time-lapse microscopy. These data sets, again, are very difficult to handle and time-consuming to manually annotate. So this makes synthetic data-driven approaches even more valuable and attractive to the biological image analysis community. And so at the same time, extending those pipelines to 3D introduce significant challenges, including more complex organelle morphologies, larger data volumes like we see in lattice light sheet microscopy, where we're getting terabyte to petabyte scale data sets, and the need for specialized 3D model architectures and tracking methods. There isn't actually a deep learning image tracking and segmentation architecture for 4D microscopy, and that's something that Aidan's working on currently. Beyond mitochondria, another very important key direction is extending this framework to multiple organelles and studying organelle-organelle interactions, such as the contact site dynamics we see in cells, such as ER mitochondria contacts, as well as, say, mitochondrial lysosome contacts or for mitochondrial degradation. We're also interested in evaluating how these models generalize across different cell types, imaging modalities like lattice light sheet, and those very, very large data sets. So we just like to end off and thank our small team. So VJ, Aidan, me, Senful, Sanjeev, that's really been behind this project for the last 2 or so years, particularly our supervisors, VJ and Senful, for pushing us to do this work and giving us like so much great feedback. Additionally, Sanjeev and Senful's group, who has been talking to us constantly from Monash, and who was responsible for collecting the data used in this project and liaising with us on that end. And of course, everyone else in VJ's Cell Systems and Mechanobiology group who joined us today. There's actually quite a number of us, so I can't list them here, but we really appreciate you guys. And we appreciate you guys for tuning in and listening, and we'd be happy to take any questions.